So this summer, not very recently, there was a new enzyme released that makes a big breakthrough in anti-aging technology. This specific enzyme is tackling one of the main aging issues, which is extracellular damage due to sugar. And it is very simple. It is not some specific epigenetic, some sort of hack. It targets, it's simple enzyme, right? It's artificial, it's not natural, but it's just a protein. It doesn't have any sort of toxic issues. It's not inherently like bad, which is very good because you don't need a lot of testing. I think it's already coming out. It's getting FDA approved and it's going over these stages where it might actually become some sort of cosmetic uh, product pretty fast, right? And it is not a small improvement. It is not some sort of irrelevant little thing. Specifically, the thing that it's like supposed to dissolve and like fix is this thing is called uh, basically these sugar things called advanced glycation end products, make which get made in your body because glucose reacts to your tissue. And there's nothing you can do. You cannot just eat some better diet to fix it. Your body always uses its own glucose either way. It's like just a natural thing that just biology just goes through because sugar is specifically just a reactive molecule. And over time, it just reacts with your tissue, forming these little matrices that cannot be dissolved. And yeah, it just like clogs stuff up, ruins your skin, makes old people have wrinkles, and it has all sorts of like even visible traits that it does. Now, this specific study is actually pretty interesting because it was not some sort of accident. It was an actually very specific planned approach. It used pretty decent technological devices and like tools. We will go over how it was actually made later on in the video, but what I want to actually cover is I just want to highlight how important this is because, as I said, this is pretty simple. It's just an enzyme, right? It looks, it's pretty new, right? It's like they just came and fixed this branch of agent, right? It's not fully fixed, but it is like a lot of it is fixed because like more than half of all these little buildups in your body kind of get cleaned up, meaning that it is not perfect, but it's pretty much a rough solution, a literal solution, just not complete. So, of course, should you be concerned about this? Should you be interested in like maybe you can buy this, maybe you can make this? How can you actually use this? Because again, like all this research is good, but it's gonna take years until like it's relevant for you for actual usage. So with this thing, you can actually like you can actually I'm sure you can actually even get it. As I said, this is simple. This is not some sort of complex thing that needs approval. It's just a protein. And what I want to know is after we get more studies, after we get more pictures, right, in the before and after, because this, if I'm not mistaken, this was done in an actual dish, in a petri dish. It was not done on a human. And there's all these little things that, of course, come into play. For example, it doesn't have immune system, right, that attacks foreign bodies, which could be an issue in human patients. There's all this little issue, basically. For example, it's also pretty big. So it's like 50,000 Daltons, which cannot pass through your skin. So, and because these damages are under your skin, right? It's not on the skin. You cannot just apply some sort of a cream. You would need to do some sort of injection or micro needling like application. But yeah, it's like again, it's not that like they didn't have that much time to actually come up with something like super. optimized. This is just like one study. It's like a prototype, pretty much. Now, does it fully solve the actual sugar glycation end products? No, of course not. Um, it is very good though for playing around. Okay, so it's actually very good to actually see some sort of differences, noticeable, measurable, visible differences, and actually, it's like a Lego brick that you can just throw it in, see how much actual age it expands. It wasn't used for aging thing. This was just simply just to prove its how it works, proof of concept. I don't think there's any studies yet because this is like what? This is like a month and a bit old, around a month before this came out. This- July 30. It came out July 14, so it's like, wait, it came out 2 weeks ago, around 2 weeks ago. So it is pretty new. It is. It came out 2 weeks ago, which is actually very interesting because everybody started talking about it. It's on YouTube. It's actually not some sort of unique thing that nobody knows about. A lot of people pick up on it. People make videos on it. It's getting popular very fast, which just shows that it's actually it's not a small thing, right? 50K accesses. That's good. So while this itself is like interesting, it's amazing, doesn't mean it's gonna be used in the whole big stack of fixing agent, okay? Because that's like another level. What it is really good though for is learning, specifically learning the approach and learning the possibilities. For the possibilities, this just shows how powerful enzymes can be. Okay? Enzymes are simply proteins that have a certain feature and they can interact with molecules. The enzyme itself, the goal of the enzyme is to find the specific fit. So you just sit down, you engineer some sort of an enzyme, and what it does, it acts as a little pattern matcher basically to find the molecule that you want. So if there's like millions and different molecules in your body. If you want to choose a specific molecule that you want to interact with, all we got to do, open like AlphaFold, open all these software, and you can engineer your own enzyme specifically for that task. Those are like little tools. They're good. And it just shows that you can pretty much tackle anything molecular that doesn't require complex DNA. Secondly, the approach is also very good. So that one's actually worth understanding. So to be specific, what they basically did is first of all, they started by saying, okay, if we're targeting sugar, let's look at an existing enzyme that exists, use it as base, an enzyme that does a similar job, because there's no enzyme that is specifically this. You need to find something that is close. And basically what they did is they picked this enzyme group that is interacting with sugars. Then what they did is they actually used all that software, all these AI models, AlphaFold 2 and so on, to actually engineer it further to do the specific task that they're trying to do. And it engineered, right? Pure software engineered an actual enzyme that does its job. It wasn't perfect. It was very weak, but they did it, right? Now, the third stage that actually was needed to like complete this was taking that weak enzyme that's supposed to do its job and giving it to bacteria and basically setting up the environment where the bacteria essentially depend on it on survival, right? So the bacteria that manage to like survive the best will have basically upgraded this enzyme into a better version. Anyways, they just ran this evolution thing of generation of bacteria, and because it's a bacteria are small, you scale it up, it actually makes an impact. And essentially, bacteria adopt this enzyme to survive. And over time, basically through mutations, they just developed better optimized versions, right? Through natural selection, literally. You can run natural selection on bacteria because there's so many of them and they divide and multiply pretty fast. So They took like what? They took like a week or two to basically run this whole evolution thing, and boom, it actually made a huge difference. It actually improved that enzyme much more. So that's the actual final product that they're presenting. Now, it is pretty interesting because the hard part of this was, I would say, the hard part is engineering that weak enzyme, that first enzyme that actually works. That's the hard part because once you actually make it, improving it is a bit easier because that's just like testing out different different ways to fold it and stuff. And eventually you're gonna come up with stuff and it's a bit better. You have a feedback loop. The hard part was actually utilizing all these AI models to make that enzyme because again, it's 50,000 daltons. That's pretty big. Okay, proteins are super complex folded structures. It's not easy to like build something that you want. Like it's very hard. There's all these physics that are going around. And the thing that actually made it happen, okay, was these models, okay, the AlphaFold and stuff. Without it, they wouldn't have done it, okay? If you actually look back, what, like 30 years or so, no AlphaFold, no nothing. You couldn't do this, okay? That's it. You couldn't do this. But now you can actually see the actual differences. So casein, collagen, ITP, hemoglobin. So yeah, so the actual damage of the sugar dissolving, it's everywhere. It's on the eye. It's everywhere. It's on your blood. Sugar is used in cells, in every cell, in your brain. It's everywhere. So the damage is everywhere, and is actually comparing how much the reversal is. Reversal, of course, is different. It is not perfect, as I said. It depends on a bunch of environmental factors. But as I said, reductions greater than 50%. That means that more than half of the issue is pretty much solved. Okay. With 7 sites showing reductions of more than 90%, which pretty much almost solved human tissues. We then saw the testability directly on human tissues. Again, this is not on humans, it's on human tissues. If I'm not mistaken, if I'm not actually lying. From 64-year-old donor, okay, looks like it's a donor, meaning that it's not an actual living human. Again, as I said, using it on actual human alive, functioning, perfectly functioning, all that stuff introduces some extra issues, but they can be fixed by just taking your time. You don't need some sort of interventions to fix extracellular matrix. Exactly. This goes into detail exactly, specifically, what sugar impacts, how it works, blah, blah, blah. But the whole idea is that all you need to know is that no enzyme, no natural enzyme, biological enzyme can actually dissolve these things. So they're kind of permanent pretty much. And that's it. Like not even some ground bacteria can do it, right? There's some ground bacteria that can actually dissolve specific Alzheimer's garbage buildup, right? Because human body cannot dissolve the specific Alzheimer's garbage, which causes Alzheimer. But there's some bacteria in the ground that can, right? With this one, there's nobody natural. So this was purely artificial. 5 rounds of evolution they did, which I don't. They don't say exactly how long it took. I think, but it should give you an idea that it's pretty fast. Definitely less than a month for sure. Why is it 5 rounds? That all it takes? Because there's millions bacteria, probably billions, and you scale it up just like in AI. You just scale up the computers, scale up the data set, terabytes, and you have a smart model. Anyways, it's pretty interesting to actually test it out. Again, everybody can access these tools. I don't think they're used like they probably use some sort of funding, but usually AlphaFold, all these most of the actual tools in the industry are free. You can access them. You don't need to pay to run them to test stuff. You can all do it yourself from home. Okay, have a Wi-Fi. That's all you need. Pretty detailed study going into a lot of stuff. Now, what about the predictions? What do I think will actually happen with this? Well, here's what I'm thinking. I'm thinking that these people will pick up on this They say, okay, we made a big thing. That's pretty good. They will focus on this specific direction. They will continue to do some other research, trying to apply similar methods, similar approaches. Mutations and previously deemed irreversible, okay? As I said, cannot be dissolved naturally. Yeah, that's about it. I might actually make a thread on this because it is interesting. If I'm making a thread on this, I'm not making it about this specific study. I'm making it about actually, specifically about advanced glycation end products, because that topic is very important and probably gonna make some summary of this. Yeah, that's about it. That's all we kind of need to know about this. It's not gonna be breaking news. It's gonna be forgotten about a month after, but it's interesting to know.